I'll be talking about IOL scope. So as we know, microscope is a boon to the science field, and it's a visual bridge connecting the macro universe to the micro universe. And it has been used in ophthalmology for various purposes like research, diagnosis, and treatment. But it's largely restricted to labs, and it's expensive, time consuming, and you require labor intensive. And so we come with point of care diagnostics. Point of care diagnostics or bedside diagnosis have the advantage of being portable, automated, has a faster processing time, reduced sample volume, and lower cost. And as we know, the goal of point of care diagnosis is to get timely diagnostics in clinics, OPDs, camps, ICUs, and operation theaters, as well as PSCs. Smartphone has been used in ophthalmology for anterior and posterior segments for various purposes. And it can be used as a point of care diagnostic. But then what about the use of it in microbiology, pathology, and parasitology? So we used this concept to make the IOL scope. So things required to make an IOL scope is a chart paper of 4 into 2 centimeters, intraocular lens of 30 diopters, 4 in number, micropore, paper puncher, and liquid adhesive. So how to prepare it? First, you have to cut out a 4 into 2 centimeter chart paper shown here. And then at one particular end, you can punch it with a puncher, a single puncher. Once you've finished punching it out, you can use one of the 30 diopter lens to be pasted at that particular hole which we created. You can use adhesive glue like Fevicol and just put one drop at each of the haptic end. Now you can see that, and keep it for drying. Once it's dried up, you can put another 30 diopter lens on top of it and attach it with the liquid adhesive. The similar manner, you can put 2 other IOLs. So totally 4 IOLs of 30 diopter lens can be placed one upon the other, stacked it up one upon the other, and then your IOL scope is ready. Once it's ready, you can use it on any of your phones by attaching it with a sellotape or a micropore. Just have to keep it aligned to the camera of the phone. So once it is attached, you're ready to go. How to use it? Source of illumination could be a smartphone, a torchlight, or the direct sun. Here, you can keep a torch on which a slide prepared can be kept. And then in this manner, you can just take photo or a video of the particular slide. These are the results. Rhizopus seen in LCB stain. This is a fungal hyphae seen on a KOH mount. Here. This is a parasite, Oystris ovis she botfly seen with the same instrument. This is a caterpillar hair. Rhinosporioidosis histopathology slide, and inflammatory cell slide, H&E stain. So early and accurate detection of microbes can be done using this instrument, and it is particularly helpful in KOH mounted fungal keratitis. It also has the ability to relate to the telemedicine for expert opinion. Also, the phone's magnification helps us increase the magnification with the digital zoom. And this is something which anybody can make it in their house. And IOLs can be used which are expired, unsterile, or which cannot be implanted into the eye. Thank you.
Amazing.
Excellent.
Amazing.
Thank you, Dr. Prithvi. Very interesting innovation there. We'll start the questions first. We'll ask Dr. Subudhi if he has any comments.
Yeah. Excellent innovative idea.
Thanks a lot.
It's very much cost effective, definitely. But my questions are there. How much magnification you achieve? Is it comparable to the ordinary microscope, lab microscope we have?
Yes, sir. Yes, sir. What I've used here is a 30 diopter lens, 4 of it. Actually, the magnification differs. Yeah, it differs according to the lenses used. And I've used 4 for 30 diopter lens, and I have compared it with the normal microscope. And we are actually doing a study with fungal hyphae detection, and we have found it to be 100%. The magnification 100x of the microscope, which we use.
How long this IOL scope stays, and how long you can use this device for? How many time? I mean, months.
Yeah, as long as the optics of the IOL is clean and clean, we can use it.
How to clean and how to preserve it? How to clean and preserve it?
You can use the same UV light which we use nowadays for Corona period. The same UV light we can just sterilize during that.
Okay, very good.
What
And
about
how
the
to get
interface?
this? Yes.
Sorry?
Yes.
Interface.
Sorry, sir?
What about
Interface?
the interface
We have
between one eye?
We have, yeah, its interface is the air interface is there. So you might actually get a little bit of artifact. But the center, you get a center artifact and just a very small one, which might distort in the central area. But most of the areas is actually very clear. As you could see in the video I showed, all the fungal hyphae were very clear. Even parasite photos were very clear. It was comparable to that of the actual microscope, and we have already done a sensitivity specificity test in which it's come up to 100% sensitivity.
Thank you, sir.
Just one suggestion: if you can make a stand for this, so that we can use any mobile, just put it on a stand where you have this IOL stack inside,
yes,
underneath, and distance also you can maintain.
Yes, sir. We have actually used a test tube holder, a holder which we use to hold the test tube. We have attached the phone to the test tube, and we are actually moving up and down and wherever we want according to the place, the site of yes.